Hello, my name is Alex Popescu, and I'm an undergraduate student in Dr. Lucia Peixoto's lab at Washington State University. I'll be talking about our work on the transcriptional dynamics of sleep deprivation and subsequent recovery sleep in the murine cortex. A lack of sleep is a widespread problem and has been shown to be detrimental to overall brain function. We also know that both sleep and sleep loss regulate gene expression throughout the brain. Nonetheless, the molecular consequences of sleep deprivation and the ability of sleep to restore baseline gene expression are still underexplored. Our goal was to provide an overview of reproducible transcriptional dynamics at the gene level across multiple sleep deprivation and recovery sleep time points. To accomplish this and also ensure that our model is both physiologically and biologically relevant, we integrated in vivo mouse data from our lab with publicly available RNA-seq data from Paul Franken's lab. In our lab, mice were sleep deprived for 5 hours and then allowed 2 hours of recovery sleep. In the Franken lab, mice were sleep deprived for 3 and 6 hours, and the mice kept awake for 6 hours were allowed 6 hours of recovery sleep. Because genes naturally change their expression throughout the day, there were time-matched controls for all experimental time points. Following RNA sequencing, salmon was used for quantification and taximeta was used to import metadata and quantification data into RStudio. The matrix of gene counts was filtered to remove very lowly expressed genes. The bioconductor packages EDA-seq and RUV-seq with a K of 6 were used for normalization. Using EdgeR, we obtained lists of differentially expressed genes for all comparison. We then performed functional annotation on a subset of these genes with David. We see that traditional normalization methods like upper quartile are not sufficient for normalizing data from across different labs. In the PCA plot on the left, PC1 explains almost 50% of the variation in the data and is the lab effect, not the experimental treatment like we would expect. After additional normalization with RUV, PC1 is the amount of sleep. Going from left to right, we have the controls, recovery sleep, and sleep deprivation. We had positive controls for 5 to 6 hours of sleep deprivation and 2 hours of recovery sleep. In these volcano plots, dots represent genes with the number of differentially expressed genes shown in blue and the percentage recovery of positive controls in red. Thanks to RUV, we were able to recover more than 90% of positive controls and also detect more differentially expressed genes, over 1,000 more in the case of RS2. After the differential gene expression analysis, we chose to focus on one meaningful subset of genes that we call fast responders. These genes are differentially expressed in the same direction after both 3 and 5 to 6 hours of sleep deprivation, but recover with just 2 hours of sleep. We made separate Euler diagrams showing the intersection of upregulated genes and the intersection of downregulated genes across all time points. We split fast responders based on whether they are up or down at both SD3 and SD56. So for example, with the upregulated fast responders, as you can see in the Euler diagram, they are the 536 genes that lie at the intersection of SD3 and SD56 only. Functional annotation revealed terms related to transcription factors like phos and kinases like SYK1 and MAP kinase. there were 785 downregulated fast responders. Looking at the list of enriched terms, it makes sense that we see ubiquitin-like protein conjugation and protein biosynthesis, since unfolded protein synthesis is known to be downregulated by sleep deprivation. Eukaryotic initiation factors, which are involved in protein biosynthesis, are examples of downregulated fast responders. Using RUV, we were able to integrate data from our lab and the Franken lab, helping us to increase our statistical power and also identify genes and patterns that are reproducible across different data sets. We were particularly interested in those genes that respond to just 3 hours of sleep deprivation, remain differentially expressed at 5 to 6 hours, and recover with only 2 hours of sleep. Although most enriched terms we identified are known to be affected by sleep deprivation and recovery sleep, it was not known that 3 hours of sleep deprivation and 2 hours of recovery sleep are enough to impact them, showing how dynamically regulated sleep is. We would like to perform functional analysis on subsets of genes that follow other patterns of recovery, such as those genes that do not recover even with 6 hours of sleep. Thank you.